Oh, hey guys, you ready? Okay, so you've all had lab. What are the two major methods for determining protein content for proximate analysis? Just say GMAS and Caldel. So we've already done Caldel, so let's talk about GMAS. Because you've all seen the instrument itself, just a quick recap that GMAS determines protein by measuring nitrogen. So it initially combusts the sample and the combustion furnace there, and then the samples move through the instrument in order to separate the nitrogen from the other gases so that it can be detected using a thermal conductivity detector. You guys probably weren't keeping a stopwatch, but that whole process took about 4 minutes per sample. So what do you think some of the advantages of GMAS over Caldel are? Lynn?
No reagent.
Yeah, what else? Megan? Yeah, because that lovely sliding block works for us now.
I can do a lot of samples that way.
That's a huge one. Amanda?
Not as much time.
Yeah, 4 minutes for one sample. Granted, as a reminder, you guys didn't see it. I'd already done it for you. I did have to calibrate it this morning. And that did take about an hour and a half to 2 hours to calibrate. And so that calibration step is something that if the instrument's not running well, it can take a little while. But once you've got it run, your throughput is a lot of samples. So from there, after the samples combusted, once again as a recap, they moved through they went into the ballast here. And in the ballast, our combustion reaction products, so our nitrous oxides and our CO2 were mixed with the help of oxygen. So oxygen is what kind of helped mix the samples in the ballast. And then they were taken, an aliquot was taken using the aliquot loop. There again, just like in GC, we don't need to send the whole amount of sample to the detector, but being able to take a smaller, so we can take a smaller amount. From there, the samples move through the reduction tube in the furnace here. And what happened in the reduction tube? Who remembers what took place there?
That's where the copper is.
What was that?
Isn't that where the copper is?
That is where the copper is. So what happened in the reduction tube?
Like nitrous changing into nitrogen.
Yeah, so the nitrous oxide species are reduced to nitrogen. Why do we need our sample as nitrogen?
That's what we're measuring.
Yeah, exactly, Molly. That's what we're actually detecting. And if you go back, review your GC notes, we talked about it in lab, how a thermal conductivity detector works. It's really important to have a pure sample, so in this case, pure nitrogen, being sent to the detector. So this instrument we call it the LECO because it's the LECO brand of the GMAS instrumentation. There's quite a few other brands that make instruments that can do the GMAS method. Tonya has another one in the building made by Buki. So these different methods can have slight variations in how the sample moves through and it's separated. So this is a schematic for one of those different example models. And the big key piece here is rather than separating and scrubbing out the components we don't need, so for example the CO2 or the water vapor, you can actually have GMAS instruments that have a GC column right in the instrumentation. So it uses that GC column then to separate your different gases from each other. So then based on when the nitrogen elutes, that is what's detected. So it's just a different variation for how we could separate and then detect our nitrogen. With that, we talked about this a little bit, but let's kind of go point by point here. Compare and contrast principle. Somebody let me know what's similar and what's different between the two methods. Claire?
They're all similar because they have the amount of nitrogen.
Yeah, what's the difference? Monique?
Dumas is like a combustion method.
Yep, and Caldwell worked by digesting the samples, but then in the detection, it's very different, right? And how we're calculating, where the Dumas measures percent nitrogen for us, where for Caldwell, we're measuring the amount of HCl titrated, and we can equate that to the moles of nitrogen that were present in our sample. Instrumentation, one thing to note, so it seems like the Dumas is a lot more automated. There are newer Caldwell instruments out there where you can have the titration being automated for you. So you can take out some of that additional human error from the Caldwell instrumentation. Also, if you have somebody, let's say at Artec or at General Mills in their analysis labs that runs Caldwell all day, every day, that is what they do, you are going to get pretty good reproducibility, they can, with that practice and that ability to kind of just really minimize their errors. Cost, which one do you think is more costly in the long run? Yeah, probably because of the maintenance of the Dumas can be very expensive. But they both also have costs in buying the units. But what's one nice thing in terms of safety and chemicals? Which one's safer?
Dumas.
Dumas, right? We don't have concentrated sulfuric acid, as Pam learned. Time, we talked about. And then applications, for the most part, are you able to use a Caldwell, do you think you can use a Dumas in the same method to measure proteins? Mm-hmm. Yeah, for the most part you can. There are a couple times when what you're measuring might not work. So for example, when we run dietary fiber, we have to correct for the residual protein in our sample at the very end. We personally don't run dietary fiber samples on the Dumas because it's called sea light. Basically, the filtering aids really kind of mess up the combustion furnace and they fill the crucible too quickly. So there we prefer to use Caldwell. So there's occasionally times, or for Caldwell, there might be some samples that work better in the Dumas, but you can, for the most part, use them interchangeably for times you want to measure crude protein. So from there, we're going to switch focuses to our research methods. So as a reminder, what type of methods are these research methods? Yeah, Julie?
Secondary method?
Yeah, they're secondary methods. And so what does a secondary method mean? Who can tell me what it means? Katie?
I mean, they are verified or validated by primary.
Yeah. So for example, when I interned one summer, they were working with products that could have very variable protein and fat content. So instead of having a Dumas or a Caldwell in there, but in order to meet their nutrition label, they had to try to, because their commodity coming in was highly variable, so they would have to tweak their formula depending. So rather than having a Dumas and Caldwell and running their samples all day, every day, what they did is they had an NIR, and they were able to calibrate it with a variety of samples over a couple days. So then they would actually send their samples out to a testing lab, like Artec, for example, at Land O'Lakes, or Gold Medal at General Mills, and they could calibrate it there. So you always need that use a primary method to help you calibrate. As a review from your IR lecture is that we would be using both near or mid IR in the food world. And how it works is it measures the absorbance based on the vibrational energy of the peptide bond. What's really nice is it's rapid. So this can be an inline method, which is something that's very advantageous when you're actually processing food in a plant. When we talk about absorbance from Monday, what do you guys remember about what wavelengths proteins absorb at? What was that?
220.
So as we're going to talk on the next slide here, or on some of these following slides, the aromatic amino acids, so tryptophan and tyrosine, absorb at 280, where the peptide bonds themselves absorb at 220. So you're getting a slightly difference, and if you go back and look, on Monday we talked about why you would sometimes choose 280 versus 220. So the second research, so now this would be a research method. Mid-IR and IR is very common in industry. It's a secondary method, but it's also widely used. So now these other secondary methods are more research methods. One of the ones we commonly use in our lab is the bicarbonate acid BCA method. So the idea behind this assay is that those peptide bonds can act as reducing agents. So specifically, in alkaline conditions, it can reduce cupric ions to cuprous ions, which gives us the reagent goes from this green color to this really pretty purple color. And so it's that purple color that we're able to measure the absorbance at. Specifically, that's 562 nanometers. Also, in addition to the peptide bonds being able to reduce, you have cysteine, tryptophan, and tyrosine that are also able to help reduce the cupric ions to cuprous ions. And the cysteine, for cysteine, is the OH group, and then, the SH group on cysteine, and then on the tryptophan and tyrosine, it would be your OH group on the amino acids that can help reduce. A disadvantage of this method is because it works by reducing the BCA reagent. If you, let's say, if you're isolating a sample, and you have to use a reducing agent to extract gluten or to extract protein, to denature the proteins to get them in solution, this method might not work as well because you're already adding reducing agents to your sample. So then you're not actually measuring the protein reducing the BCA, you're measuring whatever in your sample reducing. So when it comes to these research methods, the choice of what you use depends on your matrix. It depends on what you're familiar with. It depends on maybe how cheap the assay is, if you can buy the reagents, if you have to make your own reagents. And the book talks about that when it comes to the research methods, about why you might choose one over another. Why do we use it in our lab? Well, we mainly use it to quantify proteins in purified solutions. So for example, if I'm putting proteins in solution, but then if I'm doing a spectrophotometric method, right? So if I'm using a spec, I can't have any interference from particulates. My sample can't be turbid, so if I have to centrifuge my sample, if I take the supernatant, all of a sudden, maybe all my samples don't have the same protein in them. So if I'm correcting for the amount of, one thing I do is the extent of glycation, but I need to know how much it is per each protein. I have to know actually in each cuvette the amount of protein in my sample in order to be able to correct. So then we'd be able to use the BCA with the supernatants to determine that. So that's where it's like, and when you have purified solutions, if you need to know how much protein's actually in an individual sample, we would use something like this. How we do this is we create a standard curve. So this standard curve is BSA, so bovine serum albumin, so it's a protein that we're able to plot out at different concentrations, and then we can measure the absorbance. It's a standard curve, right? And then you have your linear equation that I'm then able to measure and quantify protein in my samples. As with all other standard curves, a big thing is you need to make sure it's linear, that your range of samples is linear, and that your actual samples you're testing for fall ideally around that middle of that standard curve. Yes? Those are my initials. So this would happen to actually be one of my samples. We do like this method because you can get a very nice R squared. And Amy and I have a competition who's had better ones. So sometimes it's the little things. From there, another type of research method, and this is the one you guys did in lab, and it's the Bradford dye binding method. Can any of you tell me how it works? What goes on there? Anybody want to give me a little recap about it?
You make a standard curve.
Yeah, you used a standard curve here too. What absorbance did you measure at?
595.
What was it?
595.
595. So what's happening here is you have your proteins, and they're able to bind this Kumasi blue dye. And that's where you see in that color change from red to blue upon binding, which then shifts that lambda max to 595 nanometers. What's nice with this one versus the BCA, the proteins aren't reducing a reagent to get color. And this instance of binding. So that might be a reason you would choose Bradford over the BCA method, is if you're worried about reducing agents affecting your readings. And who can tell me what amphoteric means?
Amanda has a question.
Yeah.
Amanda has.
Oh yeah.
No, I was just wondering, like, do all proteins bind to it? Like, what if they don't?
What, Pam, what groups are binding
I was
next?
gonna add something to what you were saying. So since it's based on the apathetic nature of the protein, that means the dye is gonna bind mostly via hydrophobic interaction.
Oh, okay.
So most proteins have that, but sometimes you don't want to use the Bradford if you're looking at protein that was not denatured and denatured proteins, because they will bind differently. The denatured protein will bind more. In that situation, you don't want to choose Bradford.
I also would be guessing if you had peptides versus intact proteins,
Yes.
Bradford would not be your best option also.
Whenever you're looking at different types of proteins, you
Mhm.
want to measure that using Bradford.
Speaking of amphoteric, can anybody tell me what amphoteric means? Yeah, Billy?
They can act as both acid and base.
Yeah. Yep. Exactly. As we said, it's sensitive and rapid, as you've seen in lab, but it's not for peptides smaller than 4 kilodaltons because of this hydrophobic bonding piece, or compatible with different detergents, because the detergents can denature the protein. So that's where that piece comes in. Yeah, Walter. What was that? Oh, amphoteric. It has the ability to act as both an acid or a base. So a couple other research methods that we didn't talk about in the notes specifically are the Burette and the Lowry method. And both of those methods actually work similar to the BCA method in that they reduce something. So you're measuring protein based on its ability to reduce an agent. The specifics are in the book if you want to look a little bit more into the detail. So if you look at this chart, we've gone through everything now except the UV at 280 nanometer absorption method. So what we have here is, as we talked about, your aromatic amino acids can actually absorb at 280 nanometers. So tyrosine and tryptophan, and then phenylalanine also can absorb. So this idea is that sometimes what we would use this, for example, is if we're measuring at a different absorbance. For example, maybe we're measuring at 517 for something. If you have the capabilities to measure both UV and visible range, you can then also at 280 see what the protein in your sample is if you have a clear solution sometimes. So then we can correct for protein right when we're reading our samples at a spectrophotometer. You can also use this as kind of it's not a very specific way, but to kind of quantify protein. You can better use it to quantify is if you did a standard curve. Or this might be used when you're using like HPLC detection. You know, the UV detector on there, you can help you identify the protein peak. What is nice is it's non-destructive. So if you're trying to kind of get some ratios of proteins using the standard curve and concentrations, you're not destroying your sample. So if you're trying to detect the protein peak in HPLC, you can collect it at 280 nanometers, and then you can use those fractions for further analysis and characterization. A disadvantage, because of the limitations of spectroscopy, is your solution has to be pure and has to be clear. To reiterate though, we talked about, so we have these peptide bonds can absorb at 220. So why would you choose 280 versus 220? There's a couple different reasons why you might choose 220 to measure the proteins at. For example, if you have small amino acids, so if you have or small peptides that you're trying to collect, and maybe those peptides don't have aromatic amino acids in there. So then you wouldn't see anything at all. So you would have to use the 220 nanometers for peptide bonds as one sample. So in general though, when it comes to mid-IR and NIR, they're actually more sensitive for assessing peptide bonds in general at this 220 nanometers, because what's the difference when we talk about what are we measuring? When we're measuring UV vis versus measuring IR, what's the differences we're measuring in the vibrations? Can you guys tell me? Yeah. Okay. So I'm thinking in terms of what's electronic, what's vibrational, what's rotational. So IR is what type, and what type is UV vis? Okay, one at a time, somebody. Sophie.
Is visible detecting the absorbance of a bond, which would be electronic because that's exciting electron, versus UV, which is measuring the stretch of a bond, so vibrational?
Yeah. Yep. Right?
Did you say UV?
IR.
UV and Viz measure.
Or IR, sorry.
Yeah.
IR is measuring
Yes.
the stretch of
There we go. So what's happening then is IR can be more sensitive than UV vis, because IR is by measuring that vibrational energy. So if you didn't want to generate, sorry, go back, last point I want to make here. If you didn't want to generate a standard curve, you could just use Beer's law to measure your absorbance, or you could also do a standard curve with proteins at various amounts. Any questions on any of this? Well, that's what I have for you, because we've already went over what you need for that. Pam?
The protein lecture from Monday, I forgot to tell Chelsea, remind her to record it, but we found, Chelsea taught the same lecture last year, and we found 2016 lecture where she stuck with the same kind of
Yep.
stuff.
Yep.
So,
I use the same notes and everything.
yeah, so I uploaded
Yep.
2016.
How do you stop?
So
Yep.
you just say,